Guys, so today we are going to talk about the MHC class 1 and class 2 molecules. So before going on to the topic, I just want to inform you that I am Agar Pathan and I study medicine in Georgia. So let's start. Hello guys. So major histocompatibility complex. So these are the proteins. These are the integral membrane proteins present on the cell surface, that is the plasma membrane, and they are synthesized by polyribosomes and are inserted in the membrane of the rough endoplasmic reticulum as a regular membrane protein. So, however, on the surface, as we'll see in the next diagram, they couple with small peptides of 10 to 30 amino acids whose origin differs depending on whether class 1 or class 2 molecules are involved. So, formation of MHC class 1 molecule. So, these are the proteins which are located on the plasma membrane of the cell, of all cells, of all nucleated cells. So, and these proteins are important for the distinction of the self from non-self cells. So how does this occur? Like the cytosolic proteins, they sit on this MHC type 1 class 1 molecules, and they are ignored by our T cells, CD. Wait. Okay, so MHC2 formation. So this is the class of peptides, sorry, proteins. They are located only on the antigen presenting cell, that is the macrophages, dendritic cell, and sometimes T cells. So the peptides that join class 2 molecules result mostly from the endocytosis and digestion in the endolysosomes. So the vesicles that contain these peptides fuse with the Golgi-derived vesicles that have class 2 molecules embedded in the membrane. So the peptide that form with the MSC protein, and as in the case of class 1 molecules, from the complex is transported to the to the cell surface, exposing the peptides to the exterior of the cell. And the main function of the MHC class 2 molecules is to present processed antigens which are derived primarily from exogenous sources to CD4 plus T lymphocyte, that is the helper T cell. And MHC class 2 molecules thereby are critical for initiation of antigen specific immune response. So yeah. So so what did I just mention, that the MHC class 1 molecule are present on all nucleated cells. Okay, all cells which have nucleus in them, but the RBCs, they, they don't have the nucleus, right? So how is the RBC not targeted by the T cells for the destruction? So with the help of this diagram, we can analyze the formation of MHC type 1 and type 2 molecules. So, as in this case, on the left hand side, we can see the this antigen associated with the MHC molecules, and T cell recognizes combination. So, this is the cytotoxic T cell, which recognizes the MHC class 1 molecule and destroys the cell via the phagocytosis, or we can say via the granzymes, or secreting perforins. So yeah. So the RBCs do RBCs are MHC1. So as we have discussed earlier, the MHC type 1 complex is only present in all nuclear cells, but the RBCs don't have nucleus, right? Mature RBCs don't have nucleus. So how do they how they are ignored by our T cells, or specifically speaking, cytotoxic T cell or killer T cell. So, RBCs do not have noticeable MHC type 1 molecules, but rather they have a higher levels of CD47 markers, which are recognized by immune system as self. On the other hand, red blood cells during the development are nucleated. They may contain multiple MHC1. Okay, so that concludes our lecture. Thank you. And if you like this video, like this video.